All right, we're going to pick back up with the remainder of chapter 27, looking at information about prokaryotes. So we're going to start off with our energy categories, and we're going to describe how they obtain their energy and also where they get their carbon from. So phototrophs are going to get their energy from light. Chemotrophs are going to get their energy from chemicals. We can further characterize these as autotrophs. So autotrophs are going to use carbon dioxide or other related compounds as their carbon source. And heterotrophs require an organic nutrient to make their organic compounds. So we're going to look at their specific energy and carbon sources on this chart here. So we want to make sure that we know what these are. So autotrophs, there are two types of those. There's photoautotrophs and chemoautotrophs. And there are two types of heterotrophs. Remember, heterotroph means other feeder. Autotroph means self-feeder. Two types of heterotrophs are photoheterotrophs and chemoheterotrophs. So back up to the top here are autotrophs. The first is our photoautotroph. This is the traditional type of plant or prokaryotic cell that's performing photosynthesis, like we would traditionally think of. The energy source is the sunlight, and the carbon source is going to be carbon dioxide or something that is related. Typically, it's going to be carbon dioxide. So examples, cyanobacteria, again, plants, and then certain types of protists like algae would fit into this category. Chemoautotrophs have a different energy source. So they use inorganic chemicals such as hydrogen sulfide, NH3, which is ammonia, and iron as the energy source, and the same carbon source as our photoautotrophs. So carbon dioxide primarily. Certain prokaryotes will fit into this category. Cephalbus is going to be an example of that. And you don't need to know the example organisms, just what is the energy source and what's the carbon source for these. For heterotrophs, our photoheterotroph uses light as an energy source and organic compounds as the carbon source. So again, certain aquatic and salt-loving prokaryotes, particularly thinking of archaeal cells. And then chemoheterotrophs, which would be the classification for us, using organic compounds as an energy source and our organic compounds as the carbon source as well. So this is a big category that would include fungi, some protists, and then of course animals as well. We also need to think about whether or not these prokaryotic cells need oxygen in the process of creating energy for their cells. So there's some different categories here. So obligate aerobes, we've talked about these in the past. They do require oxygen for cellular respiration. They are obligated to have oxygen. They die without it. Obligate anaerobes are those that are poisoned by oxygen. So they are not able to live in the presence of oxygen. They use fermentation or other substances other than oxygen for anaerobic respiration. So fermentation, you'll remember, is glycolysis with a little bit of a twist at the end. So just the very first step of cellular respiration versus anaerobic respiration, which is all three steps. But at the end, instead of using oxygen as the final electron acceptor, they use something else. So that would be more complex than those organisms that are just using fermentation. Facultative anaerobes kind of are in between these two. These are ones that can use oxygen if it's present, but if oxygen is not present, they're still able to live and carry out fermentation or anaerobic respiration. So these are really great organisms because they can utilize oxygen if it's there, but they don't die if they do not have it. Quite a few microorganisms and prokaryotes are going to perform a process that's called nitrogen fixation. And nitrogen is essential for all living organisms, primarily because it's included in amino acids. Remember, amino acids make up our proteins, and also in nucleic acids. So one piece of our nucleic acids, those are described as nucleotides. and one portion of those nucleotides are the nitrogenous bases. So nitrogen is essential for making proteins and nucleic acids, both RNA and DNA. Because they need to find a way to get nitrogen, some of these prokaryotes are able to convert atmospheric nitrogen. So again, a big chunk of our atmosphere is the nitrogen gas. They're able to convert this gas to ammonia, which is NH3, in this process, which is nitrogen fixation. So we'll watch a quick little video on this.
Microbes are the oldest form of life on Earth. They first appeared about 3.5 billion years ago and are critically important in sustaining our life on Earth today. Microbes generate at least half of the oxygen we breathe, but in 1885, the Dutch microbiologist Martinus Beijerinck discovered another critical function they performed: fixing nitrogen.
Nitrogen-fixing bacteria are absolutely essential for life because they fix nitrogen, one of the key elements, one of the key building blocks of all of our cells and all of our biochemistry. All animals and plants on the Earth depend upon it, but also human beings. The only way that we can get nitrogen is through the food that we eat that was originally fixed by the bacteria.
Although around 80% of the Earth's atmosphere is made of nitrogen, it's too stable for most plants and animals to break down. But that's where the microbes come in. Bacteria in the soil fix the nitrogen. That means they combine it with oxygen or hydrogen into compounds that plants can make use of. Some nitrogen-fixing microbes have even developed a symbiotic relationship with certain plants, like peas and beans.
Nitrogen enters the food chain as a result of microbes taking it from the air and converting it into more accessible compounds such as ammonia, which plants can then take up.
These nitrogen-fixing bacteria often live in the roots of legumes like bean plants. And the bean plants are very clever. They send out hairs from their roots that attract these bacteria in. And the bacteria essentially take up residence in the roots of these plants.
These bacteria are incorporated into root nodules. And in these special protective houses, bacteria work anaerobically to fix nitrogen into ammonia using a special enzyme called nitrogenase, which combines hydrogen and nitrogen.
In 1909, the German Fritz Haber discovered how to fix nitrogen chemically to create ammonia, leading to the production of inorganic fertilizers on an industrial scale. 1/3 of the world's population is now sustained by crops grown using artificial fertilizers. But inorganic fertilizers can be bad for the environment.
Plants that contain nitrogen-fixing bacteria can often grow in much poorer soils than other plants, so they're extremely useful for farmers. The farmers can grow the nitrogen-fixing plants, the bacteria will fix the nitrogen, and then they can come along with a plough and churn those plants into the soil and thereby enrich the soil with nitrogen. And this is one way in which nitrogen-fixing bacteria are so essential for our agriculture.
Crop rotation uses the symbiotic relationship between plants and microbes to fertilize the soil naturally. It's been practiced since ancient people began to farm. Historic rotation methods are mentioned in Roman literature and referred to by several civilizations in Asia. Today, this ancient technique has become the cornerstone of modern organic farming.
So what do these nitrogen-fixing bacteria teach us? They teach us that human beings not only require microbes, they're not only useful for us, we actually depend upon them for our survival.
So another reason to think prokaryotic cells. We also see that these microbes are able to cooperate in a cyanobacteria. So cyanobacteria called Anabaena, which is a microorganism that we're going to look at in our lab on slides. These are photosynthetic cells, and they have nitrogen-fixing cells called heterocysts or heterocytes that exchange metabolic products. So here are the photosynthetic cells of the Anabaena, and these are the heterocysts here. So again, it's interesting that they're able to cooperate and exchange materials back and forth between these cells. We talked about biofilms before. I mentioned, remember, that there are different species of prokaryotic organisms, typically bacteria, that are working together and live together in communities that coat surfaces. So things like our teeth are good examples of places that we would find biofilms. They can be problematic in some places. So this becomes a big issue with thinking about putting prosthetic limbs, prosthetic joints into people. They can become contaminated with biofilms, and that can be a major problem in people. But they are helpful for the microorganisms because they talk to each other, they exchange metabolic materials and also information about the environment so that they can all live well. So it's a very interesting process, and something that is problematic in people if they become pathogenic, but they are really great for the microorganisms. This is showing you again what we're looking at in terms of some of the examples of the groups that are found within the bacterial domain, and then some of the groups in the archaeal domain. We're going to talk about just a few examples of these and some important differences between them. So again, bacteria is the domain that has most of the prokaryotic organisms on the earth, and it is more familiar to people than the archaeal domain would be. Lots of different types of bacterial species and different nutritional types, and where we would find them living on the earth. And again, back to this, you can see that our eukarya and archaeodomains are more closely related. Bacteria is what we would describe as an outgroup. So looking at some examples of microorganisms, we're going to start by looking at proteobacteria. And there are 5 different types of proteobacteria, and we'll look at some of these examples. So main thing to remember about proteobacteria is that these are gram-negative bacteria. Remember, we talked about gram staining and gram-positive versus gram-negative, how gram-negative has 3 layers to the cell envelope, and how these will stain pink at the end of the process. Gram-positive, remember, is going to stain purple, and it has 2 layers to the cell envelope, and that peptidoglycan layer is much thicker. Some examples of the energy categories that you would find within these proteobacteria groups, these gram-negative bacteria, would be photoautotrophs, chemoautotrophs, and heterotrophs as well. Some of them are anaerobic, some of them are aerobic. Again, very wide variety of types that we'll see. So alpha, beta, gamma, delta, and epsilon are all the 5 major subgroups that we recognize currently. Alpha proteobacteria, these actually are really interesting because these are some microorganisms, some bacterial cells that are used in industrial microbiology companies to actually produce products that people are able to use. It is thought that this is the type of bacteria that was primarily the one that was a precursor mitochondria. So this is, again, this idea of endosymbiosis. So there was an aerobic alpha proteobacteria long ago that then was internalized and held on to and eventually became the mitochondria that we see in our present day eukaryotic cells. Some examples, rhizobium is a type of bacteria that we just heard about that's important in nitrogen fixation in legumes. Agrobacterium, this is used in industrial microbiology companies. It's used in genetic engineering. Something that is this particular microorganism is used for is engineering genetically modified plants. So these genetically modified plants are able to be resistant to pests. they're also able to be resistant to herbicides as well. So this is a very common way that genetically modified plants are created, by using genes from these agrobacterium species. Beta proteobacteria, another example of an organism that is found in soils, and very important in part of the nitrogen cycle as well. We can find these in water, on land, and some of these end up being pathogens for people as well. Gamma proteobacteria, a good example here is E. coli, so Escherichia coli, which again is friend and foe to us that I've mentioned before, found in the normal flora or the microbiome of mammals, including us. Generally is not normally pathogenic, becomes pathogenic if it is found in the wrong mammals. So that often can be what contaminates food products that we eat after animals are slaughtered. Some of the pathogenic groups in this proteobacteria would be Salmonella, Legionella, Vibrio cholera. These are all, again, pathogenic if they find their way into our bodies in various ways. Vibrio cholera is an example of a waterborne illness. So we often find that in places where natural disasters have happened. So hurricanes, after earthquakes, different things like that are areas where you're going to find higher proportion of people that end up with cholera. Delta proteobacteria, this includes myxobacteria, which are actually very small examples of bacteria. They are interesting because they are able to produce spores as well. We've talked about endospores and exospores and how they help microorganisms to become dormant during times when the environment is not sustainable for life. And then they can reemerge and become vegetative, we call it, able to live in the future when the environment improves. I think we haven't talked about this yet. Delovibrio, so the B is silent, Delovibrios. These are actually cannibalistic types of bacteria. So these are bacterial cells which feed on other types of bacterial cells. So that's not a problem for us, but a problem for other bacterial species. The epsilon group, most of the bacterial species that we see in this group are pathogenic as well. So Campylobacter is something that causes blood poisoning, which comes from food products. Heliobacter pylori, we have mentioned. This is the microorganism that can cause stomach ulcers. So we've mentioned that before too. So those were the proteobacteria. Again, those are gram-negatives. Chlamydia, you probably have heard of in terms of a sexually transmitted disease. So chlamydia trachomatis causes blindness if left untreated, and urethritis. It's called non-gonococcal because it's not caused by the bacteria that causes gonorrhea. But chlamydia can be treated with antibiotics if it is contracted sexually. So again, another reason to practice safe sex to prevent this transmission. So these are described as parasites because they live inside of animal cells. And this is showing you animal cells that have the chlamydia bacteria that's living inside of them. So it's this black microorganism that you can see inside of these cells. Spirochetes, these are gram-negative, but they're helical, remember, spiral in shape. Again, parasites that cause problem for us. Another sexually transmitted disease, syphilis, Treponema palladium is the name of the microorganism, the bacteria that causes syphilis, which can also be treated by antibiotics. This, if left untreated, causes all kinds of serious problems. The Tuskegee experiment was an example of major problem in how the government treated generally African-American people in the South, mostly men, were included in the study, and doctors knew at the time that these patients had syphilis, but they didn't treat them with the antibiotics that would have cured them. They let them proceed with the illness and then watch them over a long period of time to see exactly what syphilis does if you do not treat it. Eventually, it enters the brain and causes people to go crazy. Borrelia burgdorferi causes Lyme disease. So this is a type of parasite that is transmitted through ticks. So ticks actually harbor this particular bacteria. The ticks then bite a mammal and then pass the microorganism on to the mammal, and that causes Lyme disease. So that's something that people can pick up, often found in other mammals at greater degrees. So animals like deer seem to have a very high percentage of Lyme disease. Cyanobacteria, we've mentioned those previously. Gram-negative photoautotrophs, they're really important for the atmosphere because they are producing oxygen. Again, this was a very important part of the evolution of the Earth. So as we went from very low percentage oxygen in our atmosphere to very high percentages, up to the 21% that we have now, a big part of that change were these cyanobacteria, which we find very prevalently in freshwater locations, and a very big part of phytoplankton that we would see in oceans as well. So what's interesting is that, again, the plant chloroplasts that we see currently today in our eukaryotic plant cells likely evolved from cyanobacteria that were performing photosynthesis. Again, that precursor eukaryotic cell took up the cell that became the mitochondria first. That was the alpha proteobacteria. And then we know that there was a second endosymbiosis process where the cyanobacteria precursor was picked up and then converted into a plant chloroplast. So we'll be looking at cyanobacteria in our lab as well. Let's watch this. Oscillatoria. So there are some structures inside of these filamentous structures inside of the cyanobacteria, which allow for movement to take place in these cells as well. So looking at some gram-positive bacteria. So remember, gram-positive, that has a purple stain at the end of the gram stain process and has the two layers of the cell envelope. They often are found in colonies. So again, working together. Actinomycetes is an example of a gram-positive bacteria. Really important type of actinomycetes is Streptomyces, and Streptomyces is found in soils and very important in helping to break down and decompose organic matter. When you dig into the dirt, that traditional dirt smell that you're used to smelling is due to the Streptomyces that you find in the soils. Organisms that live on their own, solitary species, Bacillus anthracis is the cause of anthrax. So again, that is a pathogen. And then botulism is caused by Clostridium botulinum. Both of these, Bacillus and Clostridium, are able to create spores that allow them to live in soils and can cause disease if there is a wound or an opening in an individual. This one, anthrax, was a big problem. I don't remember the time period. I think it was like early 2000s when spores of this Bacillus anthracis were put into letters and sent to various people who, when they opened the letters, inhaled the spores. There were a few people. They were typically politicians that were sent these letters. There were a few people that actually died of anthrax from these terrorist attacks. So Streptomyces, again, Streptomyces is what we find in soils, very, very important in laboratory settings as well, because we can produce antibiotics using these Streptomyces. And then this is a human cell, and on the top of this, you can see these are mycoplasma, so very tiny little gram-positive bacteria that can be problematic in terms of being a pathogen as well. Looking at archaeal cells, so again, archaea, more similar to eukaryotic cells than bacteria, but they are prokaryotic in nature. So universal ancestor, again, is what we describe as the organism that was likely in what we would now describe as the archaeal domain. Looking at some major differences and similarities between bacteria, archaea, and eukarya. Again, you can see far more similarities between bacteria and archaea than we see in the eukarya. Mainly the absence of membrane-bound organelles is what we see in our archaea in our bacterial cells. We describe archaeal cells as extremophiles because they live in very extreme environments, as we've talked about. Some specific examples of archaeal cells would be halophiles. Halo means salt and phile means like or love. So these are organisms that live in very salty environments that would not be able to sustain life that would be of bacterial or eukaryotic nature. Thermophiles love to live in very hot environments. Here's an example again of a hot spring. This is a good example of what you would see at Yellowstone. So these can be classified, some of them are both halophiles and thermophiles, very, very hot, extreme environments.
As I'm in the midst of applying for residency right now, wish me luck, Grammarly has again been a lifesaver with my application and helping me write.
Hey, it's me, your skin. I crave being clear without irritation. CeraVe.
Looks like fun heading out into those interesting places to look for these organisms. Methanogens are another example of archaea as well. They generally live in very swampy or marshy locations where methane is produced in high rates as a waste product. Methanogens are strict anaerobes, so they are, and not all of them, but a lot of them are poisoned by oxygen. So they have to live in places where oxygen levels are going to be very low. So looking back and thinking about those energy categories, we've talked about decomposers. Remember, we described those as detritivores. Examples of prokaryotes that are performing these functions would be described as chemoheterotrophic prokaryotes. So they're breaking down dead or decaying matter or the waste products from those. And then they're very important in helping to recycle nutrients back into the environment. So these are a big part of chemical cycles or nutrient cycles, we would call them in our ecosystems. And at the end of our semester, we'll talk about those examples. So the carbon cycle, the nitrogen cycle, phosphorus, these are all examples of nutrients that need to be recycled within the environment. We've mentioned symbiosis before, so looking at how organisms interact together. Symbiosis is the ecological relationship in which two species live very close together, so they live in close contact. The larger organism is described as the host, the smaller is described as the symbiont. So the symbiont typically is going to be a prokaryotic cell. Larger organism could be multicellular organisms. There's different types of symbiotic relationships. There's mutualism, where both of the organisms benefit from the partnership. So they both get benefits from this. And we have this with quite a few of the different microorganisms that live inside and on our bodies. So our microbiome includes a lot of mutualistic relationships. Commensalism is where only one organism is going to benefit. The other one has no significant harm or help. So again, this would be a situation where maybe we have some microorganisms living inside of us. They don't really do anything positive or negative to us, but they get the benefit of living inside of us, a warm, toasty environment where they get some readily available food products. Parasitism is where the organism that is the parasite harms, so causes some sort of detriment to its host, but it does not kill its host. Now, that's where these things have to be very careful, because some parasites eventually can kill the host, but that's a problem for the parasite, right? The parasite wants to keep the host alive. We think of like worms and other types of organisms that can live inside of our bodies. They do cause detriment to the host, but ultimately they want to keep that host alive so that they can continue to live. Unfortunately, that doesn't always happen for the parasite. Sometimes that host is killed, and then that parasite has to find a new host. Parasites that end up causing diseases are described as pathogens. So we have quite a few examples of those.
The war in Ukraine forced my family to flee. My sister, mother, and grandmother left their entire
So we're in the ocean of Southeast Asia and getting ready to search for a fish that lives in a place so dangerous that most other fish won't even come near it. Ready? Let's go diving. And after a bit of searching, I finally found it. The clownfish, or anemone fish as it's also called. The beautiful tentacles of the anemone are actually deadly to most fish. The tentacles have stinging organs called nematocysts, which fire tiny darts into fish that brush by. This stinging ability protects anemones from predators and helps them capture prey for food. But how can clownfish hide in such a dangerous place? Well, that's because clownfish have a symbiotic relationship with anemones that is so important that clownfish are almost always found among anemones. Symbiotic relationships are interactions between two species that can benefit either one or both species. Symbiotic relationships are divided up into three major categories. Parasitism benefits one species while the other is harmed. Like ticks, for example. There are many examples here in the ocean. Take parasitic fish lice, for example, that cling to a host fish and feed on its body fluids. Amazingly, fish will sometimes find a cleaner fish or a cleaner shrimp to help remove the parasites from their scales, mouth, and gills. When this happens, a mutualistic relationship is formed between the fish and the cleaner organism. Mutualism is a symbiotic relationship in which both species benefit from the relationship. This bird here eats the tick, which benefits the cow. In our fish example, the host fish is clean off its parasites, and the cleaner fish gets a free meal. Both species benefit, but not the sea lice anymore. Hey, I already brushed my teeth today. The third symbiotic relationship is commensalism. Commensalism benefits one organism, but the other one is neither harmed nor helped. You might have had a bug hitchhiking on you at some point without knowing it. An example here in the ocean is when remoras hitch a ride on sharks, big rays, or turtles. The remora attaches itself with a disc on its head and then hitch a ride and feed off the food scraps that the host animal leave behind. So the sharks are not affected by the remoras being there, but the remoras get a free ride and a free meal. Sounds good to me. So back to our clownfish. Clownfish are protected from predators by living among anemones, and in exchange the clownfish chase away fish and other animals that could harm the anemone. What kind of symbiotic relationship is this? That's right, mutualistic. Okay, but we still have one important question to answer. How can the clownfish be immune to the anemone sting? Well, a clownfish must first get used to its host anemone by developing a protective mucus coating, kind of like when we use sunscreen. And there are two possible explanations for how this happens. The first explanation is that the clownfish secretes its own mucus, which lacks the substance that makes the anemone fire its stinging nematocysts. It's as if the clownfish had a protective force field or invisible cape. And the second explanation is that the clownfish uses mucus produced by the anemone. The unprotected clownfish initially touches the anemone with its fins, even getting stung slightly at first, but over time gains the protective mucus coating. Symbiotic relationships happen everywhere, even in our own bodies. Did you know that every time you eat yogurt, you're actually filling up your digestive tract with symbiotic bacteria? That's right, you become the host for these natural bacteria, and they help you process and digest the food that you eat every day. All right, so next time you go outside, stop and look around and see how many symbiotic relationships you can spot around you and record what they are. And remember, never stop exploring your world.
Here's an example from our book of mutualism. So flashlight fish lives very far below the surface. And you can see that this glowing oval down here is actually bioluminescent bacteria, which helps to provide light for the fish, but also helps it look more intimidating to other predators. We have lots of mutualistic bacteria in our GI system, as I've mentioned before. And the number of species depends on the individual. Really interesting effects of our microbiome on our mental health and on lots of other portions of our health too. So I'm going to look for some newer studies to see what are all of the effects that the microbiome is known to have on our health at this point. And then, of course, we have our pathogenic bacteria. So bacteria cause about half of all human diseases. And we know that some bacterial diseases we actually pick up from other species. So a good example is Lyme disease that we mentioned before. So ticks are the ones that actually carry the Borrelia burgdorferi, which is the spirochete here that lives and grows inside of ticks. And then ticks bite us, pass this microorganism to us. And this is a very common first sign of the Lyme disease infection, which is the bullseye rash. So if you ever see this on your body, get to the doctor very quickly to make sure that you get treated. The earlier you get the antibiotic treatment for Lyme disease, the less likely you'll suffer long-term effects from it. The pathogenic microorganisms, typically bacterial cells, can cause disease by releasing different types of toxins. So there are exotoxins and endotoxins. Exotoxins are what these microorganisms secrete and are able to cause disease, even if the microorganism that has secreted them is not present anymore. So a good example of this would be food poisoning. So there are some toxins where if you leave food out for a period of time, the microorganism grows, it secretes an exotoxin. If you were to reheat the food, you could kill the microorganism, but sometimes it does not denature the exotoxin. So you can still have illness, even though you've killed off the microorganism. Endotoxins are the toxins that are released only when the bacteria die off. Often they're components of cell walls. So when the cell walls break down, that causes the symptoms and signs of the disease in the person. We've talked about horizontal gene transfer. So we talked about conjugation, we talked about transformation, transduction. And again, this is a really common way that microorganisms can spread genes that are associated with virulence. So it's thought that pathogenic strains of E. coli have picked up these pathogenic genes that were acquired through transduction. Remember, transduction is the use of bacteriophages that move genes between either different species of bacteria or the same species as well. Bacteria, again, can be very beneficial, and it's a very common microorganism or a common species. of organism that's grown in labs. So lots of genetic engineering is done with bacterial species. And this is an example here, and this is something that could be very useful, is that bacteria are able to be genetically modified to pick up and store PHA, which is a very common component of biodegradable plastics. So this is a major issue that we're concerned with, is these microplastics that can be found in lots of different waterways and things. So these bacteria are able to pick those up. In this case, this type of bacteria can synthesize a molecule, this PHA, which could be used to make plastics that could then be eaten and broken down by bacteria. So it's a better way to recycle plastic products that we actually create. So there are bacteria that are great at bioremediation, so picking up and removing pollutants like these microplastics. There are prokaryotic cells, archaeal or bacterial, that are genetically modified to clean up oil spills. They can clean up toxic waste. So really interesting things are going on in terms of bioremediation. And this is a really great field to get into if you're interested in doing something outdoors, something to help with climate issues, cleaning up our ecosystems. So this is a great way to get involved in this process. That is it for chapter 27, and I will see you all in our next class. Thanks.